Так, і зараз ми заходимо вже в лабораторію масспектрометричного аналізу. Знову наш шеф тут, мастер-шеф. І зараз він нам буде розповідати про основні девайси, які він тут застосовує і використовує.
Okay, so this was your CMS. Now we talk about LCMS. As I mentioned, now we are in the proteomics phase. So all of these devices that are there for proteomics. So basically, all the proteomics here we do is not done. We analyze proteins, but we don't analyze it on protein level. We first break them into peptides, then separate the peptides by liquid chromatography, and as they eluting from the column, we analyze them by the mass spectrometry. So this is liquid chromatography. It will take our peptides as a mix, and it will put them on the column and separate them, and then return them small portions at a time to the mass spec, right?
So, Pavel, is it column or what is it?
Sorry?
Is it column here?
No,
or?
the column is inside the column compartment. The one thing that you need for proteomics is you need a very good reproducibility of your separations. The way to achieve that is to have you need to have column at an extremely precise temperature. And this is why you have this column compartment, and it says that it's at 60 degrees. So then from the column, as we separate the peptides, they sort of elute in groups. The better you separate, the smaller these groups. You want, in principle, in a perfect world, you would want one peptide to elute at a time. In reality, there are millions of peptides, so they elute hundreds and thousands at a time. So as they elute, they go into the mass spec, right? So the mass spec is a device that analyzes ions and analyzes them in vacuum. Okay, so as peptides elute from the liquid chromatograph, they need to be turned into ions. Those ions need to go into vacuum, and then they get analyzed by the mass spectrometer, right? So how do we convert peptides that are dissolved in liquid into separate ions in vacuum? Well, we have this thing called ionization source and electrospray ionization. Basically, as peptides elute, they get into droplets. These droplets become smaller, smaller, smaller. They break down, evaporate, and then finally we have individual peptides that fly into this hole here. And inside this device, we have vacuum now. So the process of electrospray ionization is one of the most important discoveries in chemistry, and it is one of the most famous Nobel Prizes that have been awarded for electrospray ionization, for finding out a method to get ions from liquid mobile phase into vacuum ions through an interface. So all the system we have here, they're from Thermo, and they're Orbitraps. These still, there is always a newer mass spec, but a newer, more expensive and more productive mass spec, and of course we all want newer, more expensive and more productive mass spec, but these still are fantastic systems by today's standard. So geoexactive HFX, they are all Orbitraps, and they are all high-resolution Orbitraps with very good quadrupole isolation. So this one is the oldest one. It produces fantastic data, but it has this convoluted ion path geometry that results, it's hard to clean, it becomes very, very fast, it needs many, many pumps to maintain vacuum. So as a result, over time, it's not sort of that robust. You have one pump breaking, then you have another pump breaking, you have to take it apart, you have to clean it, you have to call engineers and so on. So this device, Orbitrap 40, is a newer system. And so right now it's off. There is a little bit of a problem with it. We are waiting for an engineer to come have a look at it. So we really like this device. So you can see that if you look here, the components inside Orbitrap Explorers are the same as on HFX, but this device is like 3 times smaller. So, and as a result, because and what happened is they redesigned the positions of components, they redesigned how the pumps are attached, they redesigned all the components. Now this is an extraordinarily robust device. It produces fantastic data. Any user can take it apart and clean the parts. So this is a fantastic device. This is our main workhorse, and it produces fantastic, beautiful data. So this is Orbitrap Explorers 240. And then finally, talking about the price, this mass spectrometer and complexity. This mass spectrometer is the most complex mass spec we have because this one features what is called an ion trap. So basically what this device can do is it can isolate an ion of interest, it can fragment it, so and all of them, all of the mass specs here, they have the capability to isolate and fragment ions. But what this device can do is it can isolate a particular ion, a peptide, a metabolite, fragment it, so you break it into pieces, then it can isolate a fragment and fragment it again. So you can get more and more levels of selectivity by doing this multi-fragmentation. So so imagine how selective things are when you can isolate one of the analytes, fragment it, pick a fragment, fragment it again, and so on. So this is orbital fusion lumens. Again, it's a fantastic mass spectrometer, even though it's a little bit on the older side now. And
so this line represents how molecules are running?
No.
No.
Exactly. Well, yeah. No. This is an aesthetic choice by the company to illustrate that it's actually acquiring some data right now. So right now we have some samples here. Our collaborators, just to give you an example of what we measure here, our collaborators are interested in a particular complex, how it assembles on the DNA. So they've assembled it on the DNA and they've performed ubiquitination reaction. And they want to see if they change conditions on DNA assembly, if they change components of the complex, how the pattern of protein modifications will change. So, and this is all done in vitro on purified proteins.
So even being older or newer one, every single mass spec has to be connected to LC system, right?
Yes.
So it's next to every mass spec has to be, right?
Apart from one.